Bismillahirrahmanirrahim. Now guys, this is part 3 of chapter number 2. Comparison of the animal cell with the pre-cellular form of life. In this paragraph they have discussed simply the evolution of life, that the first cell which is evolved was a small virus, then it becomes a large virus and rickettsia. And when the conditions became normal, then they converted into bacterium, and the bacterium is then becomes the eukaryotic cells. So they have discussed the evolution of the cell, that how from the small virus it becomes a eukaryotic cell, complete cell. They have discussed in this paragraph in simple. Now the functional system of the cell, we will discuss the functional system of the cell. First of all, we will discuss the endocytosis, ingestion by the cell. So the large transport and exchange which occur through the cell membrane is by the diffusion and active transport. The diffusion which occurs, the diffusion is active, simple diffusion and facilitated diffusion. The simple diffusion occurs through the pores, through the cell membrane and through the carrier proteins. If anything which is water soluble, then the aquaporin, and then through the aquaporin they will diffuse with water, along with water. If it is lipid soluble, then the diffusion will occur through the lipid bilayer, through the interstices of the lipid bilayer. If a substance has to be carried through the facilitated diffusion, then substance will first, if this is the cell membrane and if this is the integral carrier protein, the substance will attach to this area. Yes, the substance will first attach to this, and then this compound will be carried by this carrier protein in this way to the cell. So diffusion can be occur through the pores. Diffusion can be occur through the lipid bilayer membrane directly if it is lipid soluble, or diffusion can be occurred by the carrier protein. So the diffusion has three forms. Now the active transport means that a substance goes to some area with the expenditure of energy, with the use of energy. Why it is using energy? Because the substance, if this is a barrier and this is one area and this is the other, on this side the substance is already in high concentration, and in this area it is in low concentration, but the active transport carries this substance from this low concentration to higher concentration, which is opposite to the diffusion. How? By the use of energy. So this will be discussed in the upcoming chapter in more details. So in simple words, the exchange through the cell membrane, which occurs mostly by the diffusion and active transport, but the substances which are large enough, for example the worn out cells of the body, or the bacteria, viruses, etc., how they are engulfed by the cells. So they are engulfed by the cell through the endocytosis. Endocytosis are of two types: pinocytosis and phagocytosis. Pinocytosis means the drinking of the cell. It means that a substance which is engulfed by the cell, but the substance is small. If it is small, and the engulfing of this thing is called pinocytosis. If it is large, if it is bacteria, if it is bacteria, virus, if it is worn out cell, then the engulfing of that thing is called the phagocytosis. This is the difference between the pinocytosis and phagocytosis. Engulfing of small thing is called pino, engulfing of large thing is called phagocytosis. Now we will discuss the mechanism of the pinocytosis and phagocytosis, how they are occurring. Endocytosis Endocytosis are of two types: pinocytosis, pinocytosis, and phagocytosis. Cytosis, eating small, engulfing of substance and large substances. So this is the main difference between the two, the pinocytosis and phagocytosis. How it occurs through the cell membrane. For example, if it is the cell membrane, these are actually the pitted coats. These are the coated pits. Sorry, these are the coated pits, and these coated pits contain receptors. These coated pits contain receptors, And if any substance has to go to the cell through the endocytosis, then it will first attach to this receptor. For example, it is the molecule which has to be carried through the cell. It will first attach to these receptors, and after attachment to these receptors, for example, if these molecules are attached to these receptors, then the sides of coated pits evaginate outside. Evagination eventually enclose and form endocytotic vesicles. These vesicles then eventually becomes detached from this cell membrane and goes to the cells. Eventually, the lysosomes come and the vesicle combine together, and the lysosome perform its digestive function. So, in the first stage, attachment occurs. Second, the evagination will occur, and they will enclose the vesicle. Third, they will eventually separate from the cell membrane and will goes to the cytoplasm. And then fourth step is they will combine with the lysosome and its substances will be digested by the lysosome. So these are the main four steps. Step 1, to the receptor. Step 2, to the enclosing of the vesicles. Step 3, detachment. Step 4, combination with the lysosome to form the digestive organelle of the cells. So the mechanism for both the pinocytosis and the phagocytosis is the same, but if the substance is small enough, then it is called pinocytosis. If the substances, these are the bacteria, if it is virus, if it is the wound out red blood cell, then it will be called the phagocytosis. And another thing, in the case of bacteria, if it is a bacteria, for example, if it is a bacteria, then and it is the cell membrane of the macrophage, then the bacteria, it will be dragged by this antibody, and these are the receptors for the antibody on the surface of the macrophage. Then this bacteria, through th- through this through this antibody, they will attach to the receptors on the macrophages, and this intermediation of antibody, which has dragged the bacteria to the macrophage, the intermediation of this antibody is called opsonization. What is called? I will write the intermediation of antibody between the cell, in case of every cell, if it is macrophage or any cell, the intermediation of antibody between the cell and bacteria called opsonization. This is called opsonization. Now it's done. We will discuss now the functional mechanism and characteristics of lysosomes. We have discussed the function in the second part, but and now we will discuss in more detail. The lysosome, I have told you before, the lysosome is a digestive organelle of the cell which contain hydrolysis enzymes, and every substance which has been engulfed by the endocytosis will be eventually combined with the digestive organelle of the cell, and the lysosome will drain its digestive enzyme to this vesicles and will digest the substances which and will release the usable nutrients to the cytoplasm, which are usable by the cell, and the substances which cannot be used by the cell remains in the lysosome as a residual body. Now we will take a look through the functions of lysosome. Now, for example, lysosome's function. First, bactericidal agent. Lysosome, along with the hydrolytic enzyme, it also contains an enzyme which is called lysozyme. So the lysozyme is necessary. How? Because the bacterial cell membrane, which is a substrate for the hydrolytic enzyme to act on the cell membrane and its adjacent structures. This substrate of the hydrolytic enzymes of the lysosome will not function until and cannot function until the bacterial cell wall has dissolved. So the lysozyme function to dissolve the bacterial cell wall, and after this, when this dissolution has occurred, then the hydrolytic enzyme can act on the cell membrane and its adjacent structure and can dissolve the bacteria. This is the first function of the lysosome. The second function of the lysosome is regression and autolysis. What causes, for example, what causes the placenta to come to its original normal position after the pregnancy has terminated? What causes the mammillary glands to come to its original position after the lactation period has over? What causes the tail of the tadpole to disappear? These are the changes which occur through regression, and what causes the damaging area to heal out. So the damaging area, the damaged area of the body heal out because of the autolysis. Because of the autolysis, the damaging structures are digested by the lysosomes, and their products are released for the renewing and the healing of this area. So through autolysis and through regression, functions these are the functions of the lysosome. For example, the degeneration of placenta. This regression occurs through the lysosome, mammillary glands, mammillary glands regression, and disappearance of tail of the tadpole. So these are the examples of regression and autolysis, in which the damaged area and the structures of the damaging area are engulfed and are used for the renewing and healing out of the area. This is called autolysis. Now the third function is autophagy. So what means by autophagy? Autophagy, for example, the structure which has been used for a long time, which are the old structure of the cells. So the renewing process of the old structure of the cell is called autophagy. For example, the mitochondria which are in the liver cells, they are replacing after every 10 days. After every 10 days, the liver replaces its mitochondrial pool through this autophagy. So the autophagy dissolve the old mitochondria, and the products which are released by the lysosomes are used for the regeneration of new mitochondria. So the autophagy are used for the renewing of old and worn out structures of the cells. So these are the main function of the lysosomal function: bactericidal agent, regression and autolysis, and autophagy. Now how these substances are autolysis and are renewed. First of all, if this is the cell membrane, I will show this in the book, but guys, the book you will better understand from the diagrams of textbook. For example, this is the structure which has surrounded the worn out structures of the cell. For example, mitochondria, vesicles, etc. This is called the autophagosome. In the autophagosome, when the autophagosome becomes combined with the lysosome, then it forms the autolysosome, and this is the digestive vacuole or digestive structure of the cell. And through this mechanism, the lysosome drains its enzymes to this autolysosome and digest the worn out and old structures of the cell, and the products which has been released by the lysosome is used for the renewing of these mitochondria and these structure again and again. So this is the phenomena of the autophagy. Now the function of lysosome is done. Now the functions, we will discuss the function of the endoplasmic reticulum and Golgi apparatus. The functions of the endoplasmic reticulum. We have two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum is responsible for the production of proteins. The smooth endoplasmic reticulum is responsible for the production of lipids. So these are the two main functions. The other functions of the endoplasmic reticulum is to They release date enzyme which are responsible for the glycogen, for the breakdowning of glycogen. The enzyme which are necessary, they are also released by the endoplasmic reticulum, particularly by the smooth endoplasmic reticulum, and they also provides date enzyme which are necessary for the detoxification and for the detoxification process of the cells. So they also provide these enzymes. These are the two other functions of the endoplasmic reticulum, but in particular of the smooth endoplasmic reticulum. Golgi apparatus. Functions of the Golgi apparatus. The function of the Golgi apparatus is that they receive the substances which has been released by the endoplasmic reticulum, and they pack the substance and they modify that substances. So these are the main functions. Golgi apparatus, along with the modification and the packaging of the substances, they also synthesize those substances which cannot be synthesized by the endoplasmic reticulum. For example, those carbohydrate which has some association of protein, for example, hyaluronic acid and chondroitin sulfate, etc. These are also synthesized by the Golgi apparatus. The function of hyaluronic acid and chondroitin sulfate is diversified. For example, they are present in the extracellular matrix of the interstitial spaces, and they are present in the organic matrix of the organic matrix in the bones. They are also necessary for the proliferation. They are also necessary for the migration and proliferation activities of the cells. So these are the important function of the Golgi apparatus. Now we will discuss that processing of endoplasmic reticulum secretion by the Golgi apparatus, formation of vesicles. How the substances how these substances are processed by the Golgi apparatus. So we have discussed that the substances which are released by the Golgi apparatus, they then goes, if it is synthesized by the rough endoplasmic reticulum or smooth endoplasmic reticulum, it passes through the area of the smooth endoplasmic reticulum. In these, when they pass through the smooth endoplasmic reticulum, they through the vesicles, they goes to the Golgi apparatus, and the Golgi apparatus in turn modify and package these substances and then release these substances in the form of vesicles. These vesicles then goes to inside areas of the cell or outside according to the needs. So they have discussed one example that when we bath the cells in the proteins, then after 3 to 5 minutes the proteins when we bath the cells in amino acids, then after 3 to 5 minutes the proteins are visible in the endoplasmic reticulum, and then after 20 minutes the protein become visible in the Golgi apparatus, and after 1 to 2 hours the protein are released by the cells. So this is one example they have discussed right over here. Now, types of vesicle formed by the Golgi apparatus: secretory vesicles and lysosomes. So types of vesicles, they are the secretory vesicles and they can be the digestive vesicles. For example, the Golgi apparatus in the hormonal cells, in the hormonal cells, the secretory vesicles. The secretory vesicle will contain hormones. In the neurotransmitter cells, they will contain neurotransmitters, and the enzymatically active cell, for example, the pancreas. These secretory vesicles will contain the enzymes of the pancreatic enzymes, and for example, in the macrophages, these secretory vesicles become the lysosomes. So they are the destinations of the secretory vesicles, which has been released by the Golgi apparatus. Use of intracellular vesicle to replenish cellular membrane. We have discussed that through the endocytosis and through the autophagy and autolysis, the cell membrane is constantly being degenerated. So how it is replenished? As in the case of endocytosis, when the substance goes to the cell, they carry out some of the cell membrane with itself. So in this way they are degenerated again. But if you have noticed, then the vesicles of the Golgi apparatus, which goes through the exocytosis to the outside of the cell, they are also adding the cell membrane to the cell again and again. So the cell membrane is constantly being degenerated and constantly being replenished. So in this way, through the exocytosis, through the endocytosis, the cell membrane is degenerated, but again through the endocytosis, through the exocytosis, through the vesicles of the Golgi apparatus, this cell membrane is again being replenished. So in this way they are degenerated and replenishing again and again, and this is a cycle. We will discuss the mitochondria, how the mitochondria extract energy from the nutrients. Let's start. For example, when the glucose gets into the cell, then inside the cytoplasm through the glycolysis, glucose is converted into the pyruvic acid. And the pyruvic acid then goes to the matrix of the mitochondria. And this matrix contains the enzyme of Krebs cycle, and the NAD, NFAD, NADH, and FADH. They are released by the Krebs cycle, and these NADH and FADH then are used by the oxidative enzyme which are present in these shelf-like projections or invaginations. The oxidative enzyme which are present in these are the enzymes of the electron transport chain, and the electron transport chain occur in these areas, etc. And the most abundant energy is synthesized through the help of electron transport chain, because the pyruvic acid only the two ATPs are used through the glycolysis. Four ATPs are synthesized, but the two ATP are used. So the net ATP synthesis which occurs in the glycolysis is two ATP synthesized by the processes of glycolysis. Two ATPs are synthesized by the glycolysis, and other 34 ATPs are synthesized by the Krebs cycle in electron transport chain. So for example, up to 36 ATPs are synthesized, and in this way from one glucose molecule. First of all, glucose gets entered into the cell. Glucose through the process of glycolysis is converted to pyruvic acid. Pyruvic acid then goes to the mitochondria. Through the Krebs cycle, they form the NADH and NADH. The NADH2 and FADH then through the oxidative enzymes which are present in the shelf, these carry out the electron transport chain, and the energy which is synthesized in the electron transport chain is the most abundant. And through the Krebs cycle and electron transport chain, they synthesize up to 34 ATPs. 34 ATPs. So they both synthesize 34 ATPs. These 34 ATPs and 2 ATPs which is the net gain of the glycolysis. So it makes total of 36 ATPs. From one glucose molecule, we got 36 ATPs. So now the ATPs which is synthesized by the mitochondria, there are three main important processes for which this ATP is used. These are a lot of substance is synthesized by the ribosomes through the with the help of the messenger RNA. The cell, the nucleus through its DNA signal a cytoplasm through the messenger RNA to synthesize different substances. For example, if the cell need the proteins, then through the help of messenger RNA, the ribosome will synthesize protein. If the cell needs the structural protein, it will be also synthesized in the same way. If the cell needs lipids, cholesterol, then the cholesterol will need enzymes. So the enzyme for the formation of cholesterol, fatty acid, phospholipids, etc. These all enzymes will be synthesized with the help of the messenger RNA and ribosomes. So this is the energy which is used by these processes for synthesis. Now transport across the cell membrane. The sodium-potassium channel, for example, the sodium-potassium pump, which carries three sodium to the outside and two potassium to the inside, use a lot of energy. So for the transport system across the cell membrane, most of the energy is also used by this process. Now energy used by the muscles. The myosin and actin filament which are sliding over each other uses ATP. So and everybody is using his muscles all over the day for different activities. So most of the ATP is used by the energy used by the muscles. So these are the main three metabolism, which use most of the cell's ATP. Now, locomotions of the cells. The motion which occurs through the cardiac, through the skeletal muscle, through the cardiac muscle, and through the smooth muscles. These are the obvious and particular moments, and which we are aware of these moments, which are obvious. And these muscles constitute 50% of the entire body mass. But there are two specialized moments in the body at cellular level, which we will discuss. Number one is the amoeboid moment, and number two is the ciliary moment occur in the cells. Amoeboid moment. What is the amoeboid moment? The curling line moment. This is the amoeboid essay. This is, let's suppose this is the amoeboid moment. So this curling line moments is called the amoeboid moment. This curling like amoeboid moments occur in the WBC, white blood cell, fibroblast, etc. in the amoeba. This is called amoeboid moments because this type of moment is primarily studied in amoeba. So this is why they are called amoeboid moments. Now we will discuss the mechanism of the amoeboid moments, that how the amoeboid moments occurs. Now, for example, the amoeboid moments. I will draw one cell which from any cell which perform the amoeboid moments. For example, this is the cell, and these are the pseudopodial projections of the cell. These cytoplasmic projections from the cell, these are called pseudopodia. So these projections, these pseudopodia, first of all, before performing the amoeboid moments, these pseudopodia will attach to the surrounding structures through the help of their receptors. These will attach to these surrounding structures and will fix itself. This is the leading end and this is the following end. So the leading end, what will happen? The leading end will first of all fix itself to the surrounding structure with the help of receptors, and the following end will degenerate and this degeneration of the following end will enclose in the vesicles in the form of vesicles. These vesicles will come to the leading end. These vesicles will come to the leading end and will continuously fuse with these pseudopodia and will form again and again. And in this way, the leading end is formed, regenerating again and again, and the following end is degenerating. The leading is regenerating, and the following end is degenerating. So, and in this way, the amoeboid moment is performed. These vesicles which has comes to the leading end will fuse with these pseudopodia like this. This will come to this place and will add itself to this pseudopodia, and these will fuse with this pseudopodia and will form the cell again. And the receptors which are present on the inside of this vesicles will originate to the outside and will attach to the surrounding receptor. And in this way, for example, now the cell has come to this place. Now the cell has come to this place. Again, it will send some projection to the outside, and these projections are called zoodopodia. So, and in this way, the amoeboid moment is performed. Let's take a look to the diagram of the book. In this way, this is the one pseudopodia. They have shown one pseudopodia. This is the leading end, and this is the following end. No, this following end is degenerate and is enclosed and entrapped in the vesicles. And in these vesicles come to the pseudopodia, and they then fuse with the pseudopodia, and the receptors which are present in this area is again attaching with the surrounding structure, and in this way the amoeboid moment is performed and again. But one point should be noted, that it is an active process which requires energy. How? For example, these are the vesicles which are when the vesicles are enclosing, then this is some type of contraction, and this enclosing needs energy. For example, if this is let's suppose this is a vesicles, and surrounding the vesicles are the actin filament, the contractile filaments proteins, and they associate with some other type of proteins to contract and to enclose the vesicles. So these actin and association protein needs energy, for example ATP, for its function. So it will through the help of this contractile protein, the vesicle will enclosed. When this is enclosed, and so the contractile protein will use energy, so it becomes a highly active process. Again, when this vesicle come to this projection, when these vesicles come to this pseudopodia, and it become fused with this zoopodia, and it is when it is opening to the outside, again it needs contractile proteins and energy, and in this way this is an active process. Now, types of cell which exhibit amoeboid movements, for example, macrophages, fibroblast, and other cell types, WBCs, etc. These exhibit the amoeboid locomotion. Now, control of amoeboid motion, chemotaxis. Which control the amoeboid motion? Which process control or which thing control the amoeboid motion? For example, during the sunlight, if amoeba is present in a pool, and the sunlight is increasing at the end of the day, then amoeba will start moving away from this increasing temperature to the place where the temperature is less. So in this way, this is called the negative chemotaxis. And when the amoeba are any type of cells which moves toward the stimulus, this is called the positive chemotaxis. Chemotaxis is the process by which an organism move toward or away from to the stimulus. So this is called the chemotaxis. The positive chemotaxis, moving toward the stimuli. For example, the WBCs, the WBCs during its fighting with the infection, so it moves to the area of infection. The WBCs moves to the area of infection with the help of positive chemotaxis. Why? Because this is the infection, and the bacteria and viruses release some chemicals which attract the WBC. So this is the positive chemotaxis. And in case of amoeba which is moving away from the increasing temperature of sunlight, then that is the negative chemotaxis. So the positive and negative chemotaxis. Now, cilia and ciliary movement. We have discussed the amoeboid movement. Now we will discuss the second specialized type of movement, cilia and ciliary movements. First of all, we will discuss the structure of cilia, that how the cilia is organized, and then we will discuss the mechanism of functions of cilia. Now, let's draw a diagram. For example, if this is cilia, the cilia, the central structure and the central cytoskeleton of the cilia is exoneme, and it is then surrounded by the cell membrane, the membrane which is similar to the cell membrane. So this is the structure of the cilia. Now we will discuss the structure of the exoneme. That what is exoneme actually? Exoneme, this is the central microtubules, central duplet microtubules, which is surrounded by the 9 duplet microtubules. 1 2 3 4 5 6 7 8 9 9 duplet microtubules surrounded the central duplet microtubules. These microtubules are attached by the nexine proteins. This is the protein which is called nexine. And this is the central sheet which surrounded the duplet microtubules. The duplet microtubules are connected with each other by another protein. This is the central sheet. The central sheet is connected to the duplet microtubule, to the radial spike. This is what? This is the radial spike. This is the radial spike, which connect the central sheet to the surrounding duplet microtubules. So this is the central sheet. These are the radial spike. This is the nexine proteins. Now this also contain dynein proteins, outer dynein proteins and inner dynein. These are the 9 duplet microtubules which surrounded the central duplet microtubule. The central duplet microtubules is attached to each other by proteins, and it is also then surrounded by a sheet which is called a central sheet. These duplet microtubules are connected with each other through the nexine proteins, and these duplet microtubules connected with the central duplet through the radial spike. This is the radial spike. This is the dynein protein. Dynein is ATPase containing protein. ATPase containing protein. This is the outer dynein, and this is the inner. Outer and inner. This is the inner dynein protein. Now it's done. This central tubule, which is surrounded by the 9 duplet tubules, and these adjacent protein, the central sheet, the radial spike, the dynein, this overall structure is called axoneme. This overall structure is called axoneme. Now we will discuss a little bit detail of this surrounding duplicate tubules. Now come to this page. This is the surrounding duplicate tubule, which the surrounding duplicate tubule has one A tubule and one B microtubule. The A microtubule has projections and the B doesn't have projections. The A microtubule is complete. This is a complete tubule and the B microtubule is C-shaped and it is incomplete. The A microtubule has the radial spike has attached to the A microtubule, which is in turn attached to the central sheet. This is attached to the central sheet. It also has the A microtubule also has projection. This is the inner dynein and this is the outer dynein. The inner dynein molecule is also projected from the A and the outer is also projected from the A. The one which passes inside toward the central sheet is inner dynein and the one which is outside is called outer dynein. These are the dynein molecules. This is the radial spike which attach the duplicate, the surrounding duplicate to the central duplicate microtubule. And the two microtubule has attached with each other with the nexene protein. This is the nexene. Now this is the overall structure which I have explained now. This is the central tubule, the microtubule is surrounding the surrounding structure, the nexene protein, the radial spike, the dynein, and then further detail of the duplicate microtubule. Now we will discuss some other mechanism. For example, sorry guys. We will discuss the mechanism of ciliary movement of the cilia, the forward stroke and the backward stroke. First of all, they have discussed in the mechanism, they have discussed 5 important points. First, that the cilia contain axoneme. Second, if all the structure has been removed except cilia axoneme, then cilia can still beat. If all the structures has been removed except axoneme, the cilia can still beat. The cilia can still beat. The third point is, if all the structure has been removed except axoneme, the cilia can beat if the magnesium and calcium are present. Presence of magnesium plus calcium for axoneme. For axoneme to beat, the magnesium and calcium are necessary. The fourth point is guys, during the forward motion of the cilia, the duplicate tubules which are on the front side, these duplicate tubules which are on the front side, they will move. These cilia on the front side will move outward toward the tip of the cilium, and the cilia which are on the back side, they will remain stationary, and in this way they will perform the movement. So front duplicate move and back will remain static. Number 5th point is, dynein is a ATPase containing protein. So it will use ATP. In order to carry out the motor functions of the cilia. So these were the 5 main points in the mechanism of cilia. Now we will discuss the mechanism. First of all, take a look over these 5 points. The first point is, this structure is axoneme. This is axoneme. The second is, if all the structure has been removed except axoneme, the cilia can still beat. The third is, the axoneme can beat, but the magnesium and calcium are necessary. The point is, the front duplicate will move during the forward movement, and the back duplicate, the surrounding duplicate which are on the back side, will remain static. The fifth point is the dynein is ATPase containing protein and it will use ATP to perform its motor functions. So these were the main 5 points. Now how the cilia move. Let's suppose I will show you on the hand. Let for example I will show this on the hand. This is the front surrounding tubule and this is on the back surrounding tubule. The front surrounding tubules, through the help of the dynein molecule, they will slide over the central duplet toward outside into the tip of the cilium, and the backward tubule will remain stationary during the forward movement. So in this way, these will move like this, and the back will remove stationary, and in this way the cilia will beat forward. This is the best I could do. Now guys, this is done. Now let's come to the primary and non-motile cilia. Primary serve as a sensory antennas. Non-motile, which is also called the primary cilia, these these are present as a single on one cell, and their function is acting as an antenna for the cells. So whenever through their movement, they will disturb the internal environment of the cell through the different signal transduction mechanisms. Primary cilia or non-motile cilia will disturb the internal environment of the cell, and they will coordinate the signal mechanism, or they will transmit the signal across the cell, or it can bring any changes, but the main functions of the primary or non-motile cilia is to change the internal metabolic machinery of the cell through the signal transduction, or through the opening of the channels of calcium, sodium, potassium, every. But the function of the primary and non-motile cilia is to disturb the ionic or enzymatic composition inside and outside of the cell. This is the primary cilia. For example, they have discussed one example. In the kidney tubules, they project the primary cilia is projected toward the tubules. So during the flow of fluid to the tubules, they will move and they will bring changes, or they will bring some ionic changes inside the cell. So if the primary cilia is absent, and if there is no primary cilia in the kidney, then a person will suffer from a disease called polycystic kidney disease. So it is finished now, guys. Please support my channel, subscribe, like, and comment. Thank you.